Hello and welcome to Stocks Down Under. My name is Stuart Roberts and I'm one of the co-founders of our service. And joining me from Melbourne on the morning of Friday, the 10th of October 2025 is Dr. Kilian Kelly, who's the CEO of Cynata Therapeutics, ASX CYP. Kilian, good morning.
Good morning, Stuart.
I got to hand it to you, Kilian. How did you manage to have three clinical trials about to read out major data all at once? I've never seen that in 20 years of covering life sciences.
Look, I would absolutely agree. It's pretty unusual, especially for a smallish biotech company like us. To be absolutely honest, it wasn't necessarily entirely by design. Clinical trials take a long time, as I'm sure you would be aware. If we look at the three trials that we've got going at the moment, one of them, the phase three osteoarthritis trial, is a project that really kicked off way back in like 2019. So that's really how long it takes. The GVHD trial, which is the phase two trial, which is another one that's reading out, hasn't been going quite that long, but it's still way back to sort of 2022 since we submitted the IND, for example. So these things take a long time, and the exact timelines of them is always somewhat hard to predict. So it's a little bit coincidental that the results have converged in this way, but I think it's great for us. It really creates a hugely exciting 6 to 9 months ahead for us.
Absolutely. Let's go back and paint the picture. For a long time now, we've known that mesenchymal stem cells are potent therapeutically across a range of chronic disease conditions, just because they've got such beautiful anti-inflammatory properties, amongst other things. Your company was early into that asset class, if I could put it like that. As in, Cynata was put together, if I recall, about 2008, 2009 or thereabouts.
A little bit after that, it was sort of originally about 2011 when it was originally founded.
Right, right.
Still a long
Now,
time ago now.
absolutely. Um, uh, Up till now, about 1,800 trials, clinical trials have been done in the stem cell space, focused particularly on the mesenchymal stem cells. So we've had a lot of time to understand what these cells are good for.
That's right. Look, there's been, really MSC-based research from a therapeutic point of view really got going about 20 years ago, was the first sort of initial clinical trials, and that was academic groups in Europe. Since then, it has really, exploded. As you say, there's like 1,800 clinical trials around the world have been initiated with MSCs from various sources and various entities running those trials. But the challenge, I suppose, has been not just showing that these therapies have great clinical potential, which they absolutely do, but a huge challenge that the field has faced is how do we actually make these therapies consistently and at scale? Now, that's not necessarily a huge problem if you're making a small molecule drug, because people have been doing that for, 100 years. People know how to make those products consistently and at scale. It's a well-worn path. Making cell-based therapies, which are complicated and human living cells in a consistent and scalable way is a new challenge in relative terms. And that has really been what, in my opinion, has kind of held the field back over that period of time. But we really are getting to a stage now where that challenge is being solved. And I believe Cynata's technology is the leading way to solve that challenge. And in addition, we're seeing others showing that MSCs work in phase three clinical trials and actually getting FDA approval even. And that's fantastic for the field. And I think the field, therefore, is really becoming real at this point.
Right. So the foundation technology was the ability to use induced pluripotent stem cells to produce unlimited number of cells with repeatability batch to batch. That's the competitive advantage that Cynata has long talked about. Tell us about the clinical evidence you have to date that your cells are efficacious.
Yeah, absolutely. So we have so far completed two clinical trials in two very different clinical indications. The first was in a condition called acute graft versus host disease, which is thankfully a rare condition. People may or may not be familiar with it. It's a complication of bone marrow transplantation and similar procedures, which are generally used to treat certain types of blood cancers like leukemia and lymphoma. And these bone marrow transplant procedures can be very effective in treating those cancers. But a big downside is that they can cause this condition called graft versus host disease, which results from immune cells in the transplant recognizing the recipient's body as foreign. So it's almost like those patients have somebody else's immune system inside of them attacking attacking all of their tissues and organs. And you can imagine how horrible that is.
It's fatal in many cases, right?
It absolutely is. So the initial treatment is with steroids, and that only works in about 50% of cases. In the remainder of patients, they're known as steroid resistant patients. Historical 2-year survival rates are less than 20%. So it's a really terrible condition. And unfortunately, the symptoms it causes are quite horrific in many cases as well. So it's a huge problem, albeit a rare condition in the people who get this disease. It is a huge problem. And our first in human clinical trial was in patients who had steroid resistant acute GVHD. So they had failed to respond to steroids already. And at the time, they had no other treatment options. We treated 15 patients. 13 of them showed an improvement of at least 1 grade, which is known as an overall response. And that's actually the FDA's preferred endpoint. 8 out of 15, or just over 50%, showed a complete response, meaning GVHD resolves entirely. And we saw a 2-year survival rate of 60%. Now, 60% might not sound that high, but when you compare it to what we would normally expect in this population, which, as I said, is less than 20%, it is really a very, very impressive survival rate. So we saw not just good initial efficacy,
right,
but long term.
I'm pretty sure that the p-value on that was quite low, even though there was only 15 patients in the trial.
Well, because it was actually an open label single arm trial. So we didn't actually have a control arm, which meant we couldn't do formal statistics.
Okay.
But if we were to replicate those sorts of results in the ongoing phase 2 trial that we have now, we would be very confident of showing statistically significant efficacy. So certainly those results, albeit in a phase 1 trial, we obviously have to acknowledge that that's what it was, but they were incredibly encouraging, and you won't find many trials in the literature which showed those sorts of outcomes.
Right. So we've got some great manufacturing technology. You've tested this in one of the most challenging clinical conditions one can imagine. In the meantime, you've had these 3 interesting studies where we're going to read out some data in the next potentially 6 to 9 months. The one that interests me is the osteoarthritis study. University of Sydney are running a phase 3 study. Now, admittedly, this is not under an IND, so it might be a little more challenging to take the product worldwide. But phase 3 from effectively a clinician initiated study, that was unusual.
Yeah, look, it certainly was unusual. It is unusual, but it's really been a huge opportunity for Cynata to accelerate the development of this product. And I'll just explain the background, which is that the University of Sydney, among other groups, had done a lot of early stage clinical work using MSCs from other sources to treat osteoarthritis. And they've seen lots of really encouraging results showing reductions in pain and inflammation, and even some signals of protecting cartilage. And just to explain what I mean by that, osteoarthritis is a disease where the cartilage gradually gets worn away over time. So a lot of people have this in their knee. It's very common, especially in people who get to sort of middle age and older, and their cartilage gradually gets worn away. There's currently no treatments that can stop that degeneration of the cartilage. So all existing treatments just treat the symptoms. But the data that Professor David Hunter and others had generated showed that MSCs from other sources seem to have the potential to actually not just improve the symptoms and sustain that improvement, but also to protect cartilage, which would be an absolute game changer. And so what happened is Professor Hunter approached us. He was interested in seeking grant funding to run a big trial, a randomized controlled trial with an appropriately long follow-up period to really show that disease modifying effect. And he asked us if we would be willing to supply the MSCs for that trial. So he wanted to find a kind of a commercializable, consistent supply of MSCs. So we agreed to collaborate on this project, and Professor Hunter then got NH and MRC grant funding for the trial. So it was a huge opportunity for us, simply from a financial point of view, to launch into a trial that we may not have been able to fund ourselves. But in addition to that, in discussions with the TGA, the Australian Regulatory Authority, we were also able to agree that we could use the existing data using other MSCs to support progression directly into this phase 3 trial. So it really allowed us to take this leap ahead and accelerate development. And we've also had, advice meetings with the TGA where they've told us that if the results of this trial are positive, that in principle it should be capable of supporting marketing approval in Australia. So as I said, it's been a great opportunity for us to, in a very capital efficient way, accelerate development of this product.
Absolutely. As a taxpayer, I'm very happy with that use of the taxpayers' money. What intrigues me is we're talking about cartilage coming back. Now, it's not often in the literature you see MSCs being talked about in terms of regenerating tissue of some sort, but that seems to be what's going on here.
Yeah, look, obviously we don't have the results yet, so it remains to be seen exactly what happens with the cartilage. And the previous trials that have been conducted with MSCs have typically been either not large enough or don't have a sufficiently long follow-up to really definitively answer that question. So we will find out in this trial. It is a very robust trial that is powered to show this. And what I would say is that we don't necessarily even need to show cartilage coming back. If we can simply prevent the loss of cartilage in these patients, in many cases that would be good enough, because they're moderate stage patients who still have cartilage there, and the risk for these patients is that in the absence of a treatment, it's going to keep getting worse and they're going to keep losing cartilage, and ultimately that often means they need a knee replacement, and that's clearly not something anybody wants. So even if we can just stop the cartilage loss getting worse, that in fact would be a huge success. Now, will we show regrowth of cartilage? Well, again, that would be really the holy grail. And if we do that, then it's even better. And we will find out, as I said, in the next sort of 6 to 9 months. But I think it is important to stress that that's not even what the bar for success is. If we can actually just stop this condition getting worse, that would be seen as an enormous success from a clinical point of view.
Absolutely. Another disease-modifying breakthrough coming straight out of our country. I think it's remarkable.
Absolutely. It would be absolutely huge. So, as I said, there's currently no disease-modifying therapies for osteoarthritis. So if we could be the first ever worldwide, I can't really overstate what the importance of that would be.
Absolutely. Now, you've got two other studies we're reading out. The GVHD work that we discussed earlier, that's now advanced into a later stage, and you're hoping to read out data for that next year. Larger patient numbers, and so properly powered for the success we were talking about. So let's go to kidney transplant. What are you trying to achieve there?
Yeah, so the kidney transplant trial, again, this is in collaboration with a group who have a prior interest in this area. In this case, it's based in Leiden University Medical Center in the Netherlands. And the aim here is to use the MSC product to prevent the rejection of transplanted kidneys. So as most people would probably know, if you get a kidney transplant or indeed any other organ transplant, most of the time, or almost all of the time, you'll have to take anti-rejection drugs for the rest of your life. They're called immunosuppressant drugs, and they stop your body rejecting this organ that came from somebody else.
It also weakens the rest of your immune system as well, so you tend to get a bit sick of other stuff.
Exactly. And that's the problem, right? These drugs at the moment are quite good at preventing rejection of the kidney, but what they do is they weaken the immune system, so they predispose people to serious infections, but they also actually increase the risk of various cancers. And ironically, they actually damage the kidneys. So you can imagine you've had a kidney transplant because you've had kidney failure, and you're now taking drugs that are going to damage your kidneys. It's literally the last thing that you would want to be doing. So what we're trying to do here is use the MSCs instead. So ideally, these would replace those drugs, which are called calcineurin inhibitors, or at a minimum, allow the dose to be significantly reduced. And the toxicity associated with these drugs is dose dependent. So even reducing the dose could significantly reduce the toxicity. And this study follows on from a prior trial that was run by the group in Leiden, where they used donor-derived MSCs, and they saw really encouraging results. So again, we're not sort of taking a leap into the dark here. We're sort of building on existing very encouraging data. And again, this collaboration came about through the group in Leiden kind of recognizing that if they want to really bring this therapy to the world, they need to work with somebody who has a commercializable and scalable manufacturing solution.
All right. So we've got good foundation technology. Clinical data looks good so far. We've got three shots on goal, where those shots will be taken sometime in the next 6 to 9 months. It's the stars are lining up for you, Killian. Not many investors might know about you. Now, I like to tell people you can bet on Kilian Kelly being an efficient executive. He's an Irishman, but all his education was done in Scotland, so you know how to save money, right? Um,
Indeed.
a PhD at Strathclyde, and I think you did your masters at Robert Gordon University in Aberdeen. Tell us about
That's right.
your career to date.
Yeah, so well, as you say, that you summarised my early life there. I grew up in Ireland, then went to uni, both undergraduate and all the way to PhD in Scotland. So I was there for nearly 10 years in all. And after that, then I went straight into the pharmaceutical industry. So once I'd finished my PhD, I initially started working for AstraZeneca in the London area. Obviously, most people will be very familiar with AstraZeneca, huge company. And I was working in a regulatory affairs role there initially. After that, I moved to Amgen, again, a pretty big company that I'm sure most people would be aware of. And again, this was in the London area. And I was working on the clinical development of monoclonal antibody products there, one which is actually now in the market called denosumab, which is used to treat osteoporosis and some other conditions. That was sort of my early industry career. And then in 2008, I moved to Australia. And the reason for that is I met my Australian wife in the UK, and she encouraged me to move to Australia, which, to be fair, I was quite happy to do. So
It's
I've
where
been
we
in
get all our
Australia.
best talent from.
Yeah, exactly. Yeah, I know. It's definitely a good business model for the country, I think. I've been in Australia ever since. Now, once I moved to Australia, it's a very different sector here. Those sort of big global companies like AstraZeneca and Amgen have a presence here, of course, but they don't really lead R&D from Australia generally. So I kind of switched tack and moved into the small biotech world, which I absolutely love. So initially, I worked for a consulting group for a couple of years, and that really gave me a feel for the small biotech world here. I then worked for Mesoblast, which is another MSC company, which many of your viewers may know quite well. They've had great success recently, which including getting the first FDA approval for an
Absolutely,
MSC
huge,
based
huge step forward for
therapy.
the sector.
Yeah, and that's fantastic. It's great for the MSC sector. It's great for the Aussie biotech sector, and we were delighted to see it. So I was at Mesoblast for about sort of 3.5 years. So it's quite a while ago now. I left there, I think, in 2012 or something like that. And then I had a brief stint in another Aussie biotech called Biota, and then joined Cynata in early 2014, which is really when the company got going. I mentioned earlier it was founded in 2011, but initially there was a kind of, I suppose, a struggle to get sufficient funding, as is often the case. And then in late 2013, the company was IPO'd via a backdoor listing, and Ross MacDonald, who was brought in as CEO at the time, recruited me initially as head of product development. And then I stepped up to the CEO role just over 2 years ago when Ross retired. So that's my life story in a nutshell.
Right. We'll close off the interview with one interesting aspect of the story. For a while there, the potential company maker was a partnership with Fujifilm in Japan. Now that hasn't worked out as planned. I mean, a biotech has a lot of partnerships that don't quite work out, but it impacted you for a while from a share price sense. Talk to us about what happened at Fujifilm and what your learnings from that experience were.
Yeah, so look, I should first of all say we do still have a relationship with Fujifilm. So has though changed quite significantly over the piece. So initially, Fujifilm took an option to our graft versus host disease product, so an option to the global commercial rights to that product. That was way back in I think 2017, and they invested in the company at the time. They're still a shareholder. They still own about 3.5% of the company, and they've never sold any shares. They've been diluted a bit over time, of course. But that initial partnership envisaged Fujifilm being the company that took our product forward commercially and ended up, marketing it worldwide. That didn't work out as planned, as you pointed out. So they exercised their option in 2019. Of course, 2019, we all might remember we were just about to head into COVID. We didn't know that at the time. But COVID sort of derailed things in terms of the progress of that program. One of the things Fujifilm wanted to do was move manufacturing to Japan. That was impossible because it requires a lot of travel back and forth and so on, and nobody could do that. And then by the time that had all sort of played out, Fujifilm had really changed their strategy. So it's kind of nothing to do with Cynata, but at a much higher level, Fujifilm had made the decision to become a manufacturing powerhouse in this sector, which is really what they've continued to do. So they've acquired contract manufacturing businesses, reagent businesses, cell culture, media businesses, all of that, and they're really positioning themselves as being the leading manufacturing player in this sector, as opposed to a company actually developing product itself.
Right.
And that's really what led to the shift in our partnership. So now we have a strategic manufacturing partnership, but the rights to that program came back to us. And of course, that sort of, while all this played out, there was, to be frank, a lack of progress in the clinical development and so on. And yeah, that obviously has a negative impact, but we're well past that now. That's all really well behind us. And as I said, we're very close now to our phase 2 clinical trial results, and hopefully they will be successful, and onwards and upwards.
Right. So, folks, if you haven't put Cynata on your radar screen, 3 letters, CYP. Watch this one carefully, because good things are coming potentially in the next 6 to 9 months. Kilian Kelly, thank you so much for joining Stocks Down Under.
Thank you, Stuart. Great to be here.